Welcome to Avyal Life Sciences, and with this we will be starting with our analysis of artificial sweeteners through the HPLC instrument. The process begins with an automatic sampler unit which consists of a syringe, sample tray and injector into the HPLC system, where it is carried through the stationary phase or column through tubes and pumps. The differing interactions between the sample components and the stationary phase result in varying retention times, enabling the separation of individual compounds. Our HPLC system is equipped with a UV visible detector to identify and quantify the eluted components. To determine the artificial sweetener in a food products, the mobile phase solution include a phosphate buffer. The allowed particle size in HPLC column is 0.5 and thus before adding the mobile phase in the reservoir it should be sonicated for about 10 minutes and then filtered in vacuum with a 0.45 filter paper. For the preparation of sample, firstly weigh the homogenized sample in a volumetric vial and then dissolve it in a diluent. In this analysis the diluent is milliQ water, also known as HPLC water, which has a pH of 6.999. For the sample to blend in with the diluent, it needs to be homogenized through vortex mixing, which uses centrifugal force as a principal mechanism and then sonicated for about 20 minutes in an ultrasound sonicator. After that the solution makeup is done and is then added to the centrifuge at 5000 rpm for 5 minutes. Artificial sweeteners, let us get a brief knowledge of it. Artificial sweeteners, also known as non-nutritive sweeteners, are synthetic sugar substitutes that provide the sweetness of sugar without the associated caloric content. These compounds are significantly sweeter than sucrose, often requiring only minuscule amounts to achieve the desired level of sweetness. Standard analysis of artificial sweeteners include aspartame, sucralose, saccharin, and acesulfame potassium. The HPLC technique is used to estimate the presence of these artificial sweeteners as per the FSSAI guidelines. After the analysis runtime, a chromatogram is received which provides the software different peaks for all types of sweeteners present in the sample and their quantity. This allows the candidate to assess the sample for its formulation and adulteration information.